up this Monday, and a lot to know for this test. My recommendation is you got everything memorized by Friday. Give yourself a lot of headroom to ask what-if questions, look at the learning objectives, really try to get locked in terms of the information that will be on this test. There is a supplemental lecture that's been posted on smooth muscle. There's a supplemental lecture on VAQ ratios. There's going to be, tonight I'm going to talk about how to calculate the oxygen content of blood, and then I'm just going to simply follow that up with a short supplemental lecture just to repeat what I've said today, just so that there's no ambiguity there. There's the pharmacology of smooth muscle cells. Make sure that you have a good understanding how albuterol might work on smooth muscle cells. And doxazosin. I've mentioned to you that doxazosin is an alpha-1 blocker. Really, if you know what the transduction system is, if you hear alpha-1 blocker, you should be able to predict exactly what's going to happen to smooth muscle tone. Remember, we're going to have a review session this Saturday morning at 9 a.m. I'll send out the announcement for that and the URL tomorrow. And then also the learning objectives for test number 3 will be out tomorrow. But please remember, all the learning objectives, for the most part, are at the end of the outlines every night. Let me just stop there and ask if there are any questions. Anything else we need to address? Monday night, class is going to start 5:25, and that way you got plenty of time to get logged in. More than likely, I will provide some additional minutes for this test, so hopefully that won't be an issue for anybody.
Do you have another homework assignment due?
Yes, there's a homework assignment. Thank you for mentioning that. So the homework assignment is just simply going to be over calculating the alveolar ventilation rate, calculating the oxygen content of the blood. Now, just full disclosure, I'm going to try to confuse you on this test. Now, what do I mean by that? There's going to be a long list of patient data. Concentration blood glucose, concentration sodium in the plasma, concentration potassium in the plasma, yada, yada, yada. And there might be some questions on how to calculate the alveolar ventilation rate. The data that's needed to do that will be there, but it's up to you to figure out which data you need to know and which data are there simply that aren't relevant to the question. Everybody with me on this? There's nothing there that you haven't seen. You've seen all the data throughout the semester, except now, what I want you to begin to think about is, you got a patient, you got yourself, you went to a doctor, you get all this data. What data is important, what data is not important? What do you think? It's real world. It's real world clinical data. All right, so thanks for mentioning that. But anyways, so on test number 2, I jokingly say I'm trying to confuse you. What I'm really trying to do is present to you lots of the data that we've collected over the semester, and hopefully you have the wherewithal to sift through and go, oh yeah, I need this one, I need that one, and I can calculate the answer. Okay, well, last Wednesday night, we left off talking about the O2 dissociation curve. And tonight I'm going to start off by talking about calculating the oxygen content of the blood. I already gave you a formula for doing that. We're going to start off with that formula, and then I'm going to give you some data. And you're going to try to calculate the data, or calculate the answer, given the data provided. Once we're done with that, then we're going to get into red blood cell formation, and we'll talk about the various mechanisms involved. There is this term erythropoiesis. Erythropoiesis is the process by which you're able to produce, I think, a staggering 100 million red blood cells every day of your life. That's pretty darn impressive, I think. Incredibly impressive. And that falls under the category of erythropoiesis. There's a hormone known as erythropoietin. So I'm right here, in case you're not sure where I'm at, I'm right here. So there's this hormone called erythropoietin. And it's secreted, and it stimulates the bone marrow to produce more red blood cells. Where do you think erythropoietin would be secreted from? Do you think it would be secreted from tissue that's not metabolically active or tissue that's highly metabolically active? Eric, what would you say?
That is metabolically active.
That's right, yeah, exactly. And it turns out, believe it or not, that your kidneys are the O2 sensor of the blood, because your kidneys are doing a heck of a lot of work every day. And when we get to the kidneys, I will really try to emphasize that for you. But they've got this pump called the sodium potassium ATPase pump. And it burns up a lot of energy, a lot. The kidneys are really quite amazing. Anyways, we'll talk about erythropoiesis. We're going to talk about the importance of vitamin B12, the importance of folic acid, the importance of iron, and the ability to produce new red blood cells. Then we'll talk about anemia and the definition of anemia. Once we're done with that, we're going to get into the transport of CO2 in the blood. And we'll talk about various aspects related to that. Also, tonight, you're going to be presented with a negative feedback loop for the regulation of ventilation. And I think somewhat surprisingly, at least for me, when I first learned this stuff, I would have said that we regulate our ventilation around the partial pressure of arterial O2. After all, it is oxygen that we need. And as it really turns out that under normal conditions, we regulate our ventilation around the partial pressure of arterial CO2. Mr. Bennett, why would we do that? Why would we regulate ventilation around the partial pressure of arterial CO2? Is there any chemical formula that you could think of?
Or equation?
Huh?
The Bohr equation? Or
No.
Bohr, right?
No.
Oh, cellular respiration.
Sarah Wu. Mr. Cow, can you think of an equation that would highlight the importance of CO2 and why we would regulate ventilation around CO2?
I would be guessing it is involving the equation that involve carbonic anhydrase, combining CO2 plus water to form carbonic acid with the eventuality to make bicarbonate. And that is important for
Forms
maintaining
bicarbonate ion
and
Ms. Jocelyn, you're in anatomy with Mr. Cow, right? What do you think? Does that sound right to you?
That's what I was going to guess as well.
Well, I think that's absolutely 100% correct. That is exactly right. Why would we regulate our ventilation around CO2? Because we're trying to control the concentration of hydrogen ions throughout the body. And by controlling the concentration of hydrogen ions, what are we really controlling?
pH.
Huh?
pH.
What are we really controlling?
The affinity of hemoglobin?
No. Mr. Looming, why are we interested in controlling the concentration of hydrogen ions? Can you think of like any of the first principles that you learned first night in this class?
Structure determines function.
Yeah, that's exactly right. The concentration of those hydrogen ions determines the structure of the proteins throughout the entire body, which determines their function. And we come to our fundamental principle that proteins are the machinery of cells, right? So when you put in that context, you sit there and go, wow, that's pretty cool, because now it makes it easy to understand why we would regulate ventilation around CO2. We're really not regulating ventilation around CO2. What we're regulating around is the concentration of hydrogen ions. All right. So let's get to Sean the road here. These lights are really bugging the heck out of me. I don't know if they bug you guys, but they do me. Here's our simple diagram of the 5-step model. Maybe we can have all you guys sign a petition of protest or something about the photon levels in this room. All right, all of you will tell me that step number 1 is known as ventilation. But as I've mentioned before, really what we're talking about is the ventilation rate. And there's actually a lot of stuff that goes underneath here. Make sure that you organize your notes to list all the critical things that fall under step number 1. We've got step number 2, which is gas exchange. And when we talk about gas exchange, what we're talking about is the movement of oxygen into the blood and CO2 out of the blood. So step number 2, all of you would tell me, is gas exchange. And here, under gas exchange, you're going to put Fick's law of diffusion. And that leads us to step number 3, which is gas transport. And from a fundamental principle perspective, gas transport is simple. It's the amount of oxygen per unit of blood and the rate at which we're pumping that blood throughout the body. It really boils down to 2 main factors. So gas transport is dependent upon cardiac output, that's Q dot, and cardiac output is simply the product of heart rate times stroke volume. The point I made to you before is you can pump all the blood throughout the body that you wanted to, but if there's no oxygen dissolved or bound to that blood, then the oxygen delivery rate would be 0. Alternatively, you could have a tremendous capacity to bind oxygen in the blood, but if the heart was in cardiac standstill, the delivery of oxygen to the cells of the body would be 0. It's that simple. Of course, nothing is ever simple, but. So the second factor is the oxygen content of the blood. And the oxygen content of the blood depends upon two things. The concentration of hemoglobin I'm going to come along, and underneath the concentration of hemoglobin, which I have shown before, I'm going to put something a little new. So we're going to come along here, and I'm going to put erythropoiesis. And then the second factor is the percent saturation of hemoglobin. And underneath the percent saturation hemoglobin, I'm going to put the O2 dissociation curve. And underneath that, you can put a whole variety of different factors. So here I'm going to put the O2 dissociation curve. So I've got percent saturation of hemoglobin. We're just going to go up in increments of 20%. So I start out 0, 20, 40, 60, 80, and we'll say 100%. And then down on the x-axis, we've got the partial pressure of oxygen, and this is in millimeters of mercury. Well, when the partial pressure of oxygen is 0 by definition, the percent saturation hemoglobin is 0. When the partial pressure of oxygen is 20 millimeters of mercury, I think I gave you guys a value of about 35, 37% saturation hemoglobin. Andrew, what's the P50?
27-ish.
Very good, excellent. Excellente. When the partial pressure of oxygen is 40 millimeters of mercury, we're going to say that the percent saturation hemoglobin's right around about 65, 67%. What did I give you guys before? Was that the value I gave you before, or was it 75? Nobody wrote it down?
67.
65, okay, good. All right, when the when the partial pressure of oxygen is 60 millimeters of mercury, now we're up around about 87%, 85% saturation of hemoglobin. At a PO2 of 80 millimeters of mercury, we're right around 92% saturation hemoglobin. And when we're at 100 millimeters of mercury, we're right around about 97% saturation hemoglobin. So there's our O2 dissociation curve. I've already shown you a couple things of interest related to that. We talked about the Bohr effect, we talked about the P50, but now we're focused in on actually calculating the oxygen content of the blood. In our formula for the oxygen content of the blood, so the formula is oxygen content of the blood is equal to the concentration of hemoglobin times the percent saturation hemoglobin remember, you're going to write that as a decimal. So even though I've written up there percent saturation hemoglobin, you got to make sure you understand that you're going to write that as a decimal. Times the O2 binding constant for hemoglobin. All right. So what are the units for the oxygen content of the blood? And the answer is that they're milliliters of O2 per 100 milliliters of blood. So you got to know your units. For the concentration of hemoglobin, we report that as the grams of hemoglobin per 100 milliliters of blood. So here it's grams of hemoglobin per 100 milliliters of blood. The percent saturation hemoglobin is simply reported, as I said, timed as a decimal. So I'll just put 0.xxxx. And that was good. I got red X and I've got my little red asterisk. Oh boy. Shame on me. The O2 binding constant for hemoglobin is 1.3 milliliters of O2 per 1 gram of hemoglobin. Let's come back to our diagram over here. Mandy, what's the partial pressure of O2 over here? Correct. So the partial pressure, here I'm going to put partial pressure of arterial O2 is 100 millimeters of mercury. Mandy, what's the partial pressure of O2 here? 40 what? That's correct. So here I'm going to put P subscript V, partial pressure of Venus O2 is equal to 40 millimeters of mercury. What we're going to say is that for women, yes?
I'm sorry, I can't see. Is that 4 X's? 0 point?
Yeah, the number of X's doesn't make any difference, yeah. So what we're going to do is we're going to put up on the board some standard values for the concentration hemoglobin in both men and women. Typically, you'll find that in women, the concentration hemoglobin is a little bit lower than it is in men. A couple reasons. Number one, testosterone. Number two, menstrual cycle. So here we're going to say that our textbook value for the concentration hemoglobin for women, we're going to say that it's about 13.5 grams of hemoglobin per 100 milliliters of blood. For men, we're going to say that it's 15 grams of hemoglobin per 100 milliliters of blood. All right, so we've got some data, and by the way, the concentration hemoglobin can always change because we could be dealing with somebody who's got some type of a disease that impairs or causes a reduction in the concentration of hemoglobin. We could have somebody who is competing in athletic events and decides to be a cheater and decides to take erythropoietin and inject themselves with erythropoietin and increase the number of red blood cells and by definition the amount of hemoglobin per 100 mLs of blood. How are you going to calculate this? How are we going to calculate that? Anybody, don't be bashful. I guess you're bashful tonight. Kent, how are we going to calculate that?
My guess would be when the partial pressure of O2 is at 100, the percent saturation.
All right. Let's just say that you're here. What's the percent saturation hemoglobin?
92%.
90 what?
92%.
97%, is that what you said?
Sure.
How about if you're here?
67%.
Yeah, that's right. So this value, whatever this is dependent upon where we're at in this diagram and, oh, by the way, what the partial pressure of arterial O2 is, which can change. Yes, sir.
It's an obscure question, but you know those fingertip sensors that they do for oxygen detection in the blood saturation? How do they discriminate between venous and arterial?
Good question. So what they're doing is that they're shooting a beam through your finger. Your finger is not very metabolically active. And you can do other things, but typically there's not a lot of extraction of oxygen like there might be in other places of the body. So, good place to be sampled. Hand's a pretty good place. You can take a sample of venous blood from the back of the hand. It's fairly close to the partial pressure of arterial O2. You can come along, you can warm up the hand. If you warm up the hand, you create shunts. So you get like AV shunts. So you have the arterial blood being shunted into the venous side, and so your venous blood is very arterial-like. So your friend here, this comes from this. So beneath here, I'm just going to write O2 dissociation curve. And that's right here, right? If you sit there and take a look at what I put in this diagram here, underneath the percent saturation hemoglobin, I've got the O2 dissociation curve. That's your friend. Now, one thing I do want to mention is that this region up here, I'm just going to put we'll call this the plateau, has a very important physiological aspect to it. Anybody in here skier? Do we have any skiers in here? No skiers? Come on, raise your hands. No skiers? No snowboarders? Oh, thank goodness. I was going to say, you guys are really dull. Well, to go skiing, you got to go to altitude. Where do you guys go skiing or snowboarding? Where do you go?
Mammoth.
Mammoth? Yep, been there, done that.
Fair amount.
Bear Mountain? Bear Mountain, yeah. Yeah, so Bear Mountain's and Big Bear used to be called Snow Summit. Well, actually, there's Bear Mountain. But Bear Mountain and Snow Summit are owned by the same organization, which owns Mammoth, by the way. They all used to be separate at one time. You're at altitude. Up in Big Bear, you're at probably about 7,500 feet. Mammoth, you can go anywhere from 7,000 on up, probably to closer to 10,000 feet. The higher up you go, the lower the atmospheric pressure. So you go up to Mammoth, for instance. You start here, and you go on the drive up to Mammoth, then we'll just say for the heck of it, that you're at elevation, and that leads to a reduction in the partial pressure of oxygen. Is that going to have a big impact on the oxygen content of the blood? A different way of asking that question is that going to have a huge effect? See, if we go from 100 to 80, that's a 20% reduction in the partial pressure of oxygen. How about the percent saturation hemoglobin? Is there a large change in the percent saturation hemoglobin? Help me out here. Everybody says yes, raise your hands really high. All of you sit there and say no, raise your hands really high. Glad to see that. So the cool thing is that in that plateau region, we can experience significant changes in the partial pressure of oxygen and yet have only small changes in the percent saturation of hemoglobin, which is important. This last weekend, actually until yesterday, I was up at right around 7,500 feet. Definitely can feel it, but the percent saturation of hemoglobin, I went from 95% down to about 91%. All right. So here's what you're going to do. We're going to first calculate our textbook value for the oxygen content of the blood. And I've given you everything you need to know. And by the way, we're really not interested in the oxygen content of venous blood. That's kind of like an afterthought. It's what are we taking to the cell? The venous side is what we're taking away. But what we're interested in is what are we taking? What are we bringing to the cell? So your job is to calculate for me now, what is the partial pressure partial pressure. What is the oxygen content of your arterial blood? Here's the deal though, time out. Gentlemen, I want you to calculate the normative values for women, and ladies, if you'll calculate a normative value for men, using the information up there. And we'll ask some of you for your answers. Right now, I'm kind of hung up on Santana, but don't let that throw you off. What do you guys think? You guys got an answer for me? Need more time? Maria, do you need more time? Austin. So reading that movie, what was that movie? Austin Powers, yeah. What's your answer? So your answer, if I heard you right, was 17 milliliters of O2 per 100 milliliters of blood. Anybody else, anybody have a different answer? Like, does anybody have like 16.934768, which really isn't that different from 17? That's a good value, good value. Now, listen carefully. The oxygen content of the blood is reported per 100 milliliters of blood, not per milliliter of blood. Not per milliliter of blood. So make sure you have that registered in your mind. Awesome. Yeah, go for it.
Can you repeat that? Can you repeat that?
Can I repeat what?
Can you repeat what you just said? It's not clear.
Yes, what I just said is that we're reporting the oxygen content of the blood per 100 milliliters of blood, not per milliliter of blood. If you report that per milliliter of blood, then you're going to report the wrong answer. And it will be the wrong units. Now, as it turns out, I don't know if you guys know this, but I actually can read minds. And I was reading Austin's mind, and he's going, why didn't he ask Maria? And that was a really good question. So, Maria, what are you going to tell us for a normal value for men?
18.9 or 19 milliliters of oxygen per 100 milliliters of blood.
Very good, excellent. And here's what I'm going to tell you, is that the standard textbook value is 20 milliliters of O2 per 100 mLs. You guys are around there. So that's good, that's exactly right. Now what I want you to do is calculate what's the oxygen content of the blood over here. Oop, that was good. Anybody need more time? All right. Good enough. Just to demonstrate how good I am at reading people's minds, I'm going to ask Mandy for her answer. But wait a minute, I just read your mind. You sat there and said, why is he picking on me? Why does he pick on Elizabeth?
Are we doing for men or for women?
We're going to let you do the men.
Okay. For men, I got 13 milliliters of oxygen per 100 milliliters of blood.
2 thumbs up. Ben, what do you have for women?
I have 11.75.
Yeah, that's about right. Yeah. Good, that's excellent. Very, very good. Okay, so that's how you calculate the oxygen content of blood. Not going to have you do this for this test, but here's where we're headed with all this. Is that at the end of the day, what we really care about is the oxygen delivery rate. We all know that if our cells are deprived of oxygen, they cannot produce ATP. You all learned that the first 4 weeks of this class. That was called cellular respiration, step number 5 of our model. So what is of utmost importance is the oxygen delivery rate. And the oxygen delivery rate is dependent upon cardiac output times the oxygen content of the blood. I'm just going to put it up now to show you the formula. So we're going to put V dot O2, this is the oxygen delivery rate, which is going to be in milliliters of O2 per minute, or it could be in liters per minute. So the oxygen delivery rate is equal to heart rate times stroke volume. Here we are right here. So when we talk about step number 3, gas transport, there are two primary factors: cardiac output and the oxygen content of the blood. What determines cardiac output? Heart rate times stroke volume times the concentration of hemoglobin times the percent saturation of hemoglobin times the O2 binding constant of hemoglobin. That will be on test number 4. Yes?
In your clinical setting, how do you figure out someone's stroke volume?
In the clinical setting, we're not calculating stroke volume or heart rate. To do that, you've got to be able to typically image the heart. So you can use ultrasound to image the heart, and then you can do that. You can do it through MRI imaging as well. Those are probably the two uses. I will show you what's called the Fick principle. You see, over here is Fick's law of diffusion, but there will be what's called the Fick principle, and the Fick principle has to do with that calculation right there. Anyways, there's ways to do it. All right, any questions about what I've discussed thus far? Don't be bashful. Geez, how could I come along and try to trick you? Because that's what we do. We instructors, we trick you. We don't challenge you to understand how things could change. But could come along, could change the concentration of hemoglobin. Could come along, change the shape of the O2 dissociation curve. Have you tried to figure out what the percent saturation of hemoglobin is? It's pretty simple. I mean, there's nothing rocket science about trying to think about what could he do here to kind of throw me off. Or maybe he won't. Maybe he'll just have standard things up there. All right. So we're going to come down. I'm just going to erase this part. And now we need to be thinking about erythropoiesis. Erythropoiesis is the process by which red blood cells are formed or produced. So we got the bone, and within our bone, you would tell me that there are these regions where we can form new red blood cells, and that's the bone marrow. And with our bone marrow, we have these stem cells. So there are a group of stem cells that we would refer to as hematopoietic stem cells. These stem cells can be converted into a number of different of cell types. And under this broad category of hematopoietic stem cells, we've got another group of stem cells within the bone marrow, and there's this hormone, as I mentioned earlier, known as erythropoietin. Austin, erythropoietin is what type of category of hormone?
Not sure.
Okay, speak up louder.
Not sure.
Okay. Mr. Lamb, how about you? Oh, you left. There you go. I guess she wasn't back here. Let's see here. Kathy Lay. What type of hormone is erythropoietin? If you don't know, I'm reading your mind, and you're thinking about Mr. Vargas, so that's fine. It's okay.
Doctor?
Oh, I like that. I like that a lot. You know, peptide, great guess, because you know that the majority of hormones are peptide hormones. Very good, Eric. That's the right answer. So, erythropoietin comes along and it acts on these stem cells within the bone marrow and causes them to begin to differentiate into reticulocytes, which then ultimately become red blood cells. So here I'm just going to put a dash here. It shows coming out. And now what we're talking about is we're talking about these cells coming out into the circulatory system. So actually everything I'm going to show up here, just understand it's occurring within the circulatory system. So I'm just going to put down here, we'll put differentiation. I'm just putting D-I-F-F-E-R, differentiation. And what we end up with is a red blood cell. And this red blood cell travels throughout the circulatory system for a number of days. Now here's a really interesting thing, a really interesting thing from a biological or cell biology perspective, is that these red blood cells, in their very, very early part of their life, actually have a nucleus, but they become enucleated. They get rid of their nucleus. What is in the nucleus that's so darn important? DNA are genes, right? So if you get rid of your nucleus, you no longer have the genes within that cell, and those genes code for proteins, and now you no longer have ability to make new machines. And as a result, these cells wear out. Their average lifespan's about 100 days in length. So up above here, I'm just going to put an X with a bar above it. Any of you who have had statistics, you know that an X with a bar above it is shorthand for average or mean. So the average lifespan of a red blood cell is approximately 100 days. And remember earlier, I mentioned that every day of your life, you're producing about 100 million new red blood cells, which at least from my perspective is spectacular biology. So here I'm going to put approximately 100 million new red blood cells per day. Well, when a red blood cell reaches the end of its life cycle, it's broken down. One of the primary regions or places in the body where the red blood cells are broken down is the spleen. So understand, just so I'm being clear, we make a new red blood cell, it's going to circulate throughout the body for about 100 days. At the end of that 100 days, it's no longer able to operate properly because we're not able to make new proteins. And so after that 100 days, it enters, as it's circulating through, it comes through the spleen, and there in the spleen it's broken down. Now, there's a very important commodity within these red blood cells that we definitely want to recycle. Mr. Cow, what would you tell us that is?
Iron.
It is iron, that's exactly right. So even though we're breaking down these red blood cells, what we want to do is we want to recycle the iron. Now here I am going to show the circulatory system. And it turns out that there's a protein within the blood that captures the iron. And that protein is known as transferrin. So here I'm just going to put TF plus FE. So the TF is transferrin. And simply stated, transferrin is a carrier protein that binds iron. And so now we have iron bound to transferrin, and it's circulating, and we've got this organ I'll just represent it in this little cartoon here, and above it I'm going to write liver. How many of you in here have had liver, eaten liver? Come on, raise your hands real high. So Mr. Vargas, liver's got a unique taste to it. What would you kind of describe that taste as?
Iron. Nasty.
It's got kind of what I'll call a metallic taste, but that's what I was looking for. So what happens is when the concentration of iron is high in the blood, I'm just going to erase this part. You're going to understand that the iron is bound to the transferrin. And we're just going to put that when the concentration of iron is high, then what we're going to do is we're going to store that so the iron is going to be bound to this molecule called ferritin in the liver. When the concentration of iron is low in the blood, then the iron will be released back in the blood. So it'll be released from the ferritin and causing the concentration of iron to go up in the blood. So hopefully that cartoon up there is clear. When the concentration of iron is high, we store iron in the liver. When the concentration of iron in the blood is low, the liver releases iron back into the circulatory system. Any confusion with that? And what's going to happen with the iron that's in our blood? And the answer is it's going to come along and it will be captured by new red blood cells, and so it's just cycled over and over and over again. So a very simple description of the life cycle of a red blood cell. Now there are a couple very important molecules that influence the ability to make new red blood cells. So here I'm going to put nutritional factors. That are important for having a normal concentration of hemoglobin, and that normal concentration of hemoglobin is due to having a normal number of red blood cells in the blood. First of these nutritional factors is iron. When I was up at UCLA, I had I had a number of phenomenal mentors up there, but my primary mentor actually was doing some cool stuff with the World Health Organization. This is going back into the 1970s, so a long, long time ago. And there was a country called Sri Lanka. And in Sri Lanka, their number one product that they produced was tea. So there were these escarpments where the tea pickers had to go back and forth on these switchbacks up in altitude. So they're doing a lot of work, just climbing on up and then carrying these tea leaves, these bales of tea leaves back. And as it turned out, the average hemoglobin concentration of these tea pickers was right around about 10 grams per 100 mLs of blood. So here's what I need from you right now. I need an instant description of what happened to their oxygen delivery rate. If their hemoglobin is at 10 milligrams per 100 mLs, by definition, what's happening to their oxygen delivery rate? Mr. Looney, what would you say?
It's gonna be lower.
How much lower?
Quite a bit.
Good one, I like that, very good. That's good. Let's see here, is there anybody here named Elizabeth? I can read Elizabeth's mind right now. because she's sitting there going, oh, he's asking me again. And really, you should be asking Ms. Chen. Mandy, do you have an answer for us? I'm looking for a semi-quantitative answer.
Like 30%?
How much?
30?
Yeah, that's exactly right. Very good. How would we get that? Well, we know that a normal concentration hemoglobin's 15 grams of hemoglobin per 100 mLs. We got somebody at 10. By definition, we know that their oxygen delivery rate is going to be reduced by 30%. Is 10 normal? Heck no, it's not. It's very abnormal. So what did they do? What did my primary advisor suggest? Well, the answer was to come along and provide nutritional supplements that contain iron. And sure enough, they did this. They got these workers up to 15 grams of hemoglobin per 100 mLs. And what do you think happened to their productivity in terms of going up and down those mountains? And Mr. Cowles got it. It went up because they're able to increase their oxygen delivery rate to the muscles, which are demanding a lot of oxygen, by 30%. That's a cool little story. Here's another one. Folic acid. Now, I would say in most anatomy classes, you would learn about folic acid. Mr. Cow, you had anatomy last semester, right?
Yes, along with my partner.
That's correct. Were there any other partners in it?
And a distant partner over there.
Yeah, what's color?
He's past Mr. Vargas.
Aha!
Sorry, guys.
Kent, what would you say? What is folic acid? Does that ring a bell for anybody? Raise your hand if you sit there and your anatomy instructor talked about folic acid. Mr. Bennett, what would you say?
Prenatal development.
What about prenatal development? Are we talking about like their finger, pinky? Are we talking about their
Spinal
index
cord.
finger?
Spinal cord and neural tubes.
Aha. So was there a name that went along with this abnormal development?
Spinal bifida.
Spina bifida. So what is spina bifida?
It's protrusion of the spinal cord.
Yeah, so it's not, it's a lack of closure of the spinal canal, right? So now all of a sudden, depending upon how extensive that is, that dictates how extensive the disease is going to be as a result of that. But I'm curious. So they would tell you that, oh, if you have a pregnant woman take folic acid, you're going to be able to prevent spina bifida. So for all of you in here, if you happen to be part of a team that gets pregnant, you go see your OBGYN, one of the first things they're going to sit there and say is, yeah, we want to get you on a folic acid supplement. What is it about that folic acid that's so darn important? What do you think? Andrew, what would you say? How about DNA synthesis? Because sit there and think about this. I have a stem cell that's singular, that all of a sudden becomes 2 cells, and then 4 cells, and then 8 cells. And in order to multiply, it needs to be able to synthesize DNA. So folic acid plays a key role in DNA synthesis. There's another factor, and it's called vitamin B12. Vitamin B12. Why is it important? Synthesis of DNA. Okay. So under the concentration of hemoglobin, we've got erythropoiesis, that's that story over there. A second factor, or a second aspect to this is we know that there can be instances where there is some type of disease phenomenon taking place that causes a reduction in the concentration of hemoglobin, and that's known as anemia. So take a look at this equation up here. The oxygen delivery rate, anemia, is impacting the concentration of hemoglobin. And typically, for reduction, for this definition of anemia, we're talking about a concentration of hemoglobin that's less than, Mr. Cow, do you know the answer to that? If you don't, you don't, no big deal.
I'm not sure.
12.
12, that's good, that's great. How do you know that?
I have to deliver complete blood count results to patients.
Very, very good. So we're going to put less than, we'll just put 12 grams of hemoglobin per 100 milliliters of blood. Well, what can cause this reduction in the concentration of hemoglobin? Number one, you could have a decrease in what we'll call the red blood cell count. In other words, the number of red blood cells per unit volume is less than normal. So I'm just going to put decreased red blood cell count with normal amount of hemoglobin per cell. And of course, if you have a reduction in the number of red blood cells per unit volume, but each red blood cell has a normal complement of hemoglobin molecules, you're still going to have a reduction in the concentration of hemoglobin in grams of hemoglobin per 100 mLs of blood. Well, we could have a normal red blood cell count with a reduction in the amount of hemoglobin per cell. And then we could have a combination of 1 and 2. So that's, hopefully that makes sense to you. That's pretty easy. All right, so if we're talking about a reduction in the number of red blood cells, that could be due to something like hemorrhaging. I'm just gonna put HEM for hemorrhage. By the way, just as an interesting side note to this first one, is that we had a technician here, lab technician, this is going back many, many years, probably two decades now, and she had been menstruating for 30 days straight. Why did she go see a doctor? Because she felt like, you know what, CRAP. And so she goes to the doctor, her hemoglobin levels way down, and so they get her on some drugs, and next thing you know, miraculously, she's feeling great again, and she has a normal hemoglobin concentration. So you can have things like hemorrhaging or slow, constant bleeding. How about if we had some nutritional factors? For instance, you had a decrease in vitamin B12, or you had a decrease in folic acid, normal levels of vitamin B12, normal levels of folic acid. So you could have nutritional factors. Make sure you put that down there. Here's a real interesting one, because you can have what's called hemolytic anemia. Here we could be talking about sickle cell anemia. We could be talking about so-called thalassemias. Sickle cell anemia is really fascinating. If anybody's had a class in cell biology or genetics, this is a classic lesson there, because here you have what's called a point mutation, a single point mutation. You have basically the substitution of one amino acid in the globin gene. So remember, hemoglobin is at the core of it. It's got these 4 subunits of the alpha subunit for hemoglobin and the beta subunits. And so in sickle cell anemia, you have a single point mutation, a substitution of one amino acid, and that leads to a rather dramatic consequence. So here is a cell. And that cell, where are we at in this diagram? The answer is we're down here. I don't have it labeled, but we're talking about step number 5. We're talking about the cells. And all of you would sit there and say that in terms of our 5-step model, what we're really talking about is the ability of the cell to use oxygen to make ATP. And of course, that happens in the mitochondria. How's the oxygen getting to the cells? Well, the left ventricle is pumping the blood out into the aorta, and then from the aorta into the arterial tree, and then from the arterial tree into capillaries. And here's a capillary at the tissue level. And here's our red blood cell. And we've got hemoglobin, and bound to the hemoglobin is oxygen. So as I've done before, I'm just showing an asterisk between these two molecules to indicate that they're bound to one another. When oxygen is bound to hemoglobin, that's known as oxyhemoglobin. And normally what would happen is that as this red blood cell, this oxygenated red blood cell, transits past the cell, some of the oxygen will go into the cell and then into the mitochondria. And here's our molecule of hemoglobin. It no longer has oxygen bound to it. And so this is referred to as deoxyhemoglobin. This is under normal conditions. And the critical point in this diagram is that the morphology, the structure of our red blood cell doesn't change as it transits past these metabolically active cells, as it gives up the oxygen. But how about in somebody who suffers from sickle cell anemia? That's actually a good term up there, sickle cell. The red blood cell changes its shape, its structure, when it unloads oxygen. So in sickle cell anemia, what happens is when our red blood cell comes past metabolically active cells and unloads oxygen, it actually changes its shape. It sickles. And as a consequence of the sickling, these red blood cells become sticky. And they form a blood clot. That's no bueno. That's no good. Because, Mr. Lamb, what happens to the flow of blood past this metabolically active cell? Should be really obvious to you. It's not a trick question. So we've got this clot. What happens to the flow of blood past the cell? Say louder. It stops, right? If we've got a clot and it's blocking the blood vessel, then the movement of blood past that cell stops. Mr. Lamb, because the flow of blood past this cell stops, what happens to the delivery of oxygen to that cell? What happens to the oxygen content? The oxygen content right here, since the flow of blood stops. The cell doesn't stop being metabolically active, so the flow of blood stops and gets stuck right here, and we continue to suck oxygen out of the blood as much as we can, but what's going to happen to the partial pressure of oxygen with the flow of blood stopping? Is it going to stay at 100 millimeters of mercury? Is it going to go higher, or is it going to go lower? Miss Elizabeth.
Lower.
And every second that goes by, it's going to continuously get
Lower.
lower. That's right. So because of this clot, the formation of this clot leads to what is called ischemia. One of the most important terms in human biology. Different way of saying it, physiology. Different way of saying it, cell biology. Mr. Cow, what is the definition of ischemia?
Hypoxia due to lack of blood delivery.
So you guys heard what he said, right? He said ischemia is a reduction in blood flow. That's what you said, right? That's what he said. Ischemia, a reduction in blood flow. Now, what was the first part of what you said?
Hypoxia.
Hypoxia, 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 hypoxia. Hypoxia, hypoxia.
That's right.
That means what?
Lack of oxygen.
It means reduction in oxygen, right? Hypoxia. Hypo, less than oxygen. So this leads to hypoxia. Ms. Camilla. Hypoxia can lead to
Heart attack or stroke.
no, that's correct. Can lead to injury of any cell in the body. But what's the worst form of hypoxia?
I'm not sure.
Mr. Vargas?
Death.
How about anoxia? A means without, right? So the worst form of hypoxia is anoxia. Anybody in here, tell me what's the most powerful pain that a human being can experience other than the loss of a loved one?
Migraine, maybe? I don't know.
No, not even close.
Childbirth.
That's a good one. Very good. That's excellent. I've never had anybody say that, but I really like that. It's a good one. Yeah, that is funny. funny and true. How about ischemic pain? Ischemic pain is in the category of one of the most powerful pains that a human being can suffer. So up here in our diagram, what we're going to do is we're going to come along and understand that when we say ischemic pain, what are we really saying? It's not the lack of blood flow. It is, but that's not what's really creating the pain. It's the lack of oxygen that's creating the pain. But understand that ischemic pain, yeah, that pain arises because there's a reduction in blood flow, which means there's a reduction of the delivery of oxygen. That's what it really means. Everybody with me on this? So here we're going to put ischemic pain. Very, very, very powerful form of pain. Very powerful. Here's the sad thing, is that for a lot of individuals who suffer from sickle cell anemia, they have these, I'll call them episodes or attacks, where they've got this tremendous amount of ischemic pain. How are you going to help them out? Are you going to give them a vitamin C? Or are you going to sit there and say, hey, here's some Advil, take some Advil? How about some Tylenol? Sorry, those won't touch it. So you got to dip into a whole new category of drugs that you really don't want to try to administer, but you're going to have to. Mr. Cowan, what type of drugs are we going to talk about? Talking about ecstasy?
Thrombolytics or heparin?
You could, but that's not going to get rid of that pain. Morphine, we got to get them going on morphine. And here's what happens is these people, in some cases, are in this downward spiral where you start using morphine, well, guess what? You get addicted to it. And so to touch the pain, the next time you need more morphine. And the next time you need more morphine, and you're turning these individuals who are otherwise normal, you're turning them into drug addicts. And of course, this isn't a thing that applies to all these individuals who suffer from sickle cell anemia, but certainly for some of them, is they end up dying not because of sickle cell anemia, but because of drug overdose, right? Sorry to take you guys over so much. Real story, my wife used to be a pediatric nurse. I shouldn't say used to. She wants me to say that she's a retired pediatric nurse. And she was working over here at Irvine, Irvine Medical Center. And she had a girl that was 7 years old. And her mom suffered from sickle cell anemia. And now this little girl was starting to develop ischemic pain. And here's what she asked my wife. She goes, am I going to become like my mom? Because she knew that her mom was addicted to morphine. And you can imagine as a little kid, for any one of us, that would be a bit of a horror story for sure. Okay, I've said enough. Let's take a